Hi everyone, it's Adon here. Just wanted to recap with you all what we did today, talk about the end of activity 3 and then 4, and then talk to you guys about line graphing, which is activity 5 that we didn't get to in lab. We've been out of time today, but you guys are still expected to know this information, even though it's not required of you to put in your lab notebook since we didn't get to it in class. I would highly suggest that you do put it in your lab notebook because it is something that you could be potentially quizzed or tested over, and I would hate for you guys not to have those notes. So as you watch this, I would take these notes just to make note of the things that you guys will be responsible for knowing. So I want to make sure that we go back to activity 3. If you guys recall, during the Zoom meeting, I did the activity with you guys. Turns out I did part of it wrong, so I have redone it to make sure that I explain it to you correctly and so that you guys actually understand. So the top part with the 3 bullets, if you'll see my cursor, this is all correct. So the explanation that I did there was fine. 1.89 liters is 1,890 milliliters, and it is also 0.00189 kiloliters. And if you recall, I used this acronym to get there, but you can also do powers of 10 and conversions. Feel free to ask me to explain that to you guys as well. I can go over it with you in office hours or over email. The part that I did wrong was the calculation of if I needed 5 liters of 2% milk, how many containers would I need? So I went back and set it up the right way that is just easier and makes more sense. I set it up as a formula. So I have 1.89 liters and I need 5 liters. So 1.89 liters times a random, so I made it C, you could do X for a random variable, but I made it C for containers. So 1.89 liters times a random amount of containers will give you 5 liters. So we need to figure out how many containers of 1.89 liters will give you 5 liters, if that makes sense. So to recap again one more time, you have 1.89 liters, you want to know how many containers of 1.89 liters will give you 5. So what you do is you just solve this equation that you've set up. So what I did is I divided by 1.89, so you end up with the containers equals 5 liters over 1.89 liters. I divided that out. I got 2.6 containers and I round that up to 3. So if I needed 5 liters of 2% milk, I would need 3 containers for those 5 liters. This is the correct way to do it and the way that you guys should be practicing. I would recommend you set up an equation so you don't get mixed up like I did when I did these calculations with you guys. But feel free to always go back through and check your work. If something doesn't make sense, go back through and like re-multiply it out and see if it's actually a logical answer. So that's that for this activity. Let's move on to activity 4. So if you remember, we talked about the scientific method, and here's a little flow chart on the scientific method. And this PowerPoint will also be up in iLearn without my video if you would like to go over that again. But basically the steps of the scientific method is that you ask a question, you do some background research, then you form a hypothesis, which is basically an educated guess based on your background research to answer your question. Then you set up and you do your experiment to test your hypothesis. Once you do the experiment, you analyze the data that you've collected of that experiment, and then you draw a conclusion. And then once you have that conclusion, you can then communicate your results. So for activity 4, what you guys were supposed to do, we did part of this in class, was that you were supposed to get into your breakout groups and to design a controlled experiment to test your hypothesis for the question, how does height affect how far a person can jump from a standing position? So let's say I'm doing this question. So I'm here, I'm asking how far can a person jump from a standing position? Does that correlate to height? So my hypothesis is that the taller you are, the further you jump. So that's my educated guess to answer this question, is I think that taller people can jump further than shorter people. And I would like to come up with something to test that hypothesis. So in order to come up with an experimental design to test your hypothesis, I need to sit down and to figure out how to do this. So I'm 5'7", 5 feet 7 inches. So I just would go through and I'd be like, okay, so I'm 5'7". This is how much 5'7" is in meters. Don't know it off the top of my head, but you can look up the conversion. So I'm this many meters tall. And then I would pick a spot in my room, and let's say I jump. So I measure from the beginning. So I measure where I stand to start to jump, and then I jump, and then at the end I measure where I finished, and then I measure that distance. Also, yes, then I measure that distance. In order for an experiment to have like results that are well communicated, I recommend that you do something more than once. So if I jumped one time and my height was 5'7", I compared it to somebody else who jumped one time and their height was like what, 5'4", and it was just one data set, one sample for each person. That's fine. It gets you the answer that you need. It's just a higher probability of your answer maybe being wrong because you don't have as many sample points. So if I was doing this experiment, I would say, okay, I'm in my breakout group. Let's do that. Everybody jumps 3 times and you average the length of your jumps. So I jump once, I measure how far I jumped. I jump again, I measure how far I jump. I jumped the 3rd time, I measure how far I jump. I add them all up, I divide by 3, I get the average. That average will be my data point for my height. So that way it's a little bit more averaged out for the amount of jumps. So then each person ends up with an average data point based on their height, and then we put those in the table. So their heights will be in one, and then the distance that they jumped will be in the other. Then we can look at the results. So some things to think about, if that's the experiment I set up, some things to think about was whether or not my experiment was actually a controlled experiment. So a controlled experiment is scientific testing done under controlled conditions. And so there's obviously, if you guys remember what we talked about in class, some things that I just didn't control for. So basically there were confounding variables in my study. Confounding variables are extra variables that you didn't account for in your experiment. So some confounding variables in this experiment would say, like, I didn't tell my breakout group that we should do everything barefoot, that I should measure my height from barefoot instead of from if I was wearing platform shoes, because the shoes you're wearing differs. I told my group to all jump 3 times, so that was standardized, which was good, because if I jumped once and someone else jumped twice or 3 times and somebody else jumped 4 times, then the data wouldn't be the same. So that was standardized in my experiment. Obviously, my group members and I wouldn't have the same type of floor available. I have hardwood, but someone might have tiles, someone might have carpet, someone might be outside, and the type of floor influences the amount of spring you've got and how far you can jump, so that's a confounding variable. And then just in general, this is human biology, so the way you are feeling as a person also affects your data. So like if you're tired today, you won't jump as far. If you're sore, your muscles might hurt, so you won't jump as far. If you have more energy than someone, then you might be putting more energy into your jump, which means you would jump further. So these are just a few confounding variables. There's definitely more to think of for this experiment in particular. And so when we set up experiments in the future, keep in mind to try to standardize as many things as. So now that we've looked at standardizing our experimental design and making sure we don't have any confounding variables, let's move on to how we look at the data from an experiment. So what we've got right now is we have in our Excel sheet, you should have written your heights of your group and then the distance that everyone jumped from your group, right? So you have your own personal group data. So by looking at a table like that, it's hard to visualize to see the trend of your results. So what we do in order to see the trends of our results is we graph them. So graphing is a very good way to see your trends and your results. I would like you to take a moment right now, pause this video, go over the line graphing resource page that's in iLearn. And that will help you kind of understand a little bit more of how to graph. Basically, so some important things to think about when creating a line graph. You guys are going to be graphing by hand, so this is why your lab notebook should be graphing paper, just because it's easier to graph by hand if you've got the little boxes. Make sure that you title your line graph, that you have a title for it. And it should mention both your independent and your dependent variables. So each axis should also be labeled with a title. So the line graphing resource page has an example table and an example graph of how you would turn that table into a graph. So I would look at the resource page, and then I would try and look at your table in, I blinked, in the Excel sheet and see if you can turn that table into your own line graph. So we've got some terms to make sure that you guys understand. So the difference between the independent variable and the dependent variable are very important, and they differ based on which axis they go on your line graph. So the independent variable is the variable that is controlled or changed in a specific experiment. So in our experiment, we were looking at whether or not, well, actually, you know what, let's wait. Let me define the dependent variable first. So the independent variable is the variable that you change, that you are in control. You are the one that changes that specific variable. The dependent variable is the variable that changes as a result of the independent variable. So it's the variable that you're measuring. So in our experiment, we are looking at whether or not height correlates with the distance you jump. So height is the independent variable because it is the variable that you are controlling. So the height doesn't depend on the distance. It's the opposite way around, right? So the distance that somebody jumps depends on how tall they are. So when you change the height, it changes the distance that someone jumps. So that's how you look at the dependent versus independent variable. If that's confusing, ask me, make sure you let me know. I'll help you figure it out a little bit more. So for a line graph, make sure that the x-axis, this axis, the one that's right here, is the independent variable. And then the dependent variable, the y, so like this is my camera split. If this is your axis, the y-axis should be your dependent variable. So make sure that you set that up correctly when you are looking at making your line graph. So also, always make sure that your axes are equal. So if you've got a data point that's 1 and then you have another data point at 35, you can't do like one box be 1 and the next box above it be 35 and the next box above be like 43. You have to standardize it. So each box has to be like 5, 10, 15, 20, and then your points will go where they go. But you have to make sure that your axes, that each axis is equal. And you don't have to start at 0. So if all your data points start at 30, there's no point in going and starting at 0 and going all the way up to 30. Just start at 30. And that's totally fine to start, like to have your first tick on your axis be a number that isn't 0 as long as it increases in equal increments after that. That's the main point to take away from this. So try graphing your data table set, or try graphing the sample data table on the line graphing resource page, and then check your answer with the graph that they have on there and see if you can get that down. This was, oh yes, I totally forgot. So this is actually a graph of, in the lab manual, there's a sample table. So you can go to the lab manual, use that sample table, try to turn that table into a line graph, then check yourself with this one. And this is roughly what your line graph should look like based on the table that's in the lab manual, if you don't want to use the table in our Excel sheet data. Make sure you come to me if you have any questions with this. So after we figure out how to graph our lines, that's basically it for the experiment. The last bit of it was just to make sure that you knew how to graph. Yeah, so make sure that if you want to, you can go through the lab manual and look at activity 5 and see what we were going to do with it. If you would like to go through and do the experiment, I'd be happy to go over it with you if you're interested with that. And these are some wrap-up questions that we didn't end up getting to, but I think it's helpful to look at these questions and to try to think about the answers. So especially with biases and underrepresented individuals, that's important with human biology and human research. So take a second, think of any biases that might have been in the design of your experiment, just things that you weren't aware, or if there's individuals that are underrepresented or that couldn't have done this experiment and what that says about your data. And if there are issues with this, a lot of biology research, it's especially human research, it's hard to make sure that every individual is represented. But if you take a sample and you do an experiment on a sample, like let's say you did an experiment on our class, and let's say it turns out that the taller you are, the further you can jump based on our class. If our class isn't representative of the population of the United States, then we can't say this is true for everyone. And this becomes a problem if you have underrepresented individuals. So individuals of varying heights, individuals of varying ethnicities and racial backgrounds, individuals of varying ability. That's something that we didn't really think about in the design of this experiment. What if you can't jump? What happens then? And what does that mean if you are not represented in this sample? So these are just some smaller things to think about and to ponder for your experiment. And for experiments in general. Please let me know if you have questions or if you'd like to talk through these questions with me. And then just the announcements and reminders from lab. Make sure that you have everything that you need to complete for next week, and I will see you then. Thank you guys. Let me know if you have any questions over this video or anything that we did in lab.